Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is molecular basis of inheritance. And in the previous lecture, we have already talked about the Human Genome Project. Today, we are going to talk about one of the interesting topic that is the DNA fingerprinting. So, dear students, in Human Genome Project, we have studied that 99.9% nucleotide bases are exactly same in all people. How much? 99.9%. The nucleotide bases in the DNA are similar in all people. So only 0.1% differences is there in nucleotide sequence between the different organisms. And in spite of that, you can see this many different faces, millions of faces. Okay. And each face is different from the another individual. So why? Because there is a certain unique sequences in each individual, and by use of this certain unique sequences we can separate or we can determine one individual as a separate individual from the another one. And to identify or determine this nucleotide sequences, the technique which is used, it is known as DNA fingerprinting. So today we are going to talk about this DNA fingerprinting. So dear students, if we talk about this DNA fingerprinting, this DNA fingerprinting is also known as DNA typing or DNA profiling. And this DNA fingerprinting, if we talk, it is a technique that is used to determine the certain specific nucleotide sequences of DNA which are unique to each individual. So it is a technique used to determine unique nucleotide sequences which are unique to each individual. Okay. Now, so DNA fingerprinting can distinguish one human being from the another with the exception of the monozygotic twins. Monozygotic twins cannot be separated by this DNA fingerprinting because they have almost similar genome. And we know that 99% of bases among human is same. 99.9%. 99.9% nucleotide base sequence is same in all people. So how much difference is that? Only 0.1% of genome, or 3 into 10 raised to 6 base bases base pair difference is seen among individuals. So the differences does occur not only in genes but also in the repetitive DNA. We remember differences which are unique to the any individual, or this 0.1% differences. This differences the differences occur not only in genes but also in repetitive DNA. Now, DNA fingerprinting is involved in identifying differences in some specific region in DNA sequence that is known as repetitive DNA. So, DNA fingerprinting so DNA fingerprinting, if we talk, this involve identifying differences among individuals in some specific regions in DNA sequence called repetitive DNA. Now it is known that this repetitive mean DNA means if ATGC sequence is there, if ATGC is repeated many times, ATGC, then it is known as repetitive sequences. Now it is known that repetitive DNA are separated from other bulk genomic DNA as a different peak, as different peaks during density gradient centrifugation. The bulk DNA, if we talk about the peak, the bulk DNA form major peak and other small peak are referred as satellite DNA. Okay, such way these repetitive sequences are separated in a density gradient centrifugation. Now, these repetitive sequences which are present in these repetitive sequences which are referred as a satellite DNA, this repetitive DNA of a satellite DNA they are classified into the two categories based on base composition, that is ATGC is either it is AT rich or GC rich. After second base composition, the second is length of segment, and third number of repetitive units. So based on these three particular categories, the satellite DNA is classified into two categories. The first is VNTR. Now VNTR stands for variable number tandem repeats. Now we remember tandem repeats means those sequences which are repeated immediately. Means if ATGC is repeated immediately after the ATGC, then it is known as the tandem repeats. And this VNTR or variable number tandem repeat is also known as mini satellite. Whereas second is SSRs, that is single sequence repeat, or STRs, that is short tandem repeats. And this short tandem repeats or SSR, it is also known as microsatellite. Now, why it is known as microsatellite? Because the length of this microsatellite, a repetitive sequence, which is a microsatellite or single sequence repeat, is with length 1 to 6 base pair. Means the length of the microsatellite or the single sequence repeat or short tandem repeat like repetitive sequences is 1 to 6 base pair. Means if it is a AAA is repeated, so it is SSR. If 80 is getting repeated, it is small microsatellite. If ATGC is getting repeated, means immediately then it is a, we can say, SSR or microsatellite. Whereas mini satellite, if we talk, this mini satellite are varies from size varies from zero point 0.1 to 20 kilobase pair. Kilobase pair means 1000 base pair. So zero point 0.1 to 20 kb length is of VNTR is found. Now after this, it is known that this VNTR or mini satellite are surrounded by conserved restriction sites, and the sequence is arranged tandemly or immediately or repetitively in many copy number. Now it is known that copy number means if they are in large copies, means ATGC is copied 1000 times, then 10,000 is the copy number. Now this copy number varies from chromosome to chromosome in an individual. And this is known that this number of repeats this number of repeats show very high degree of polymorphism. Okay, so this is what a VNTR and SSR. Now it is known that this repetitive sequences or this sequences, these repetitive sequences do not code for proteins. They are not going to code for any proteins. Okay, but they form large portion of human genome. But they form large portion of human genome. And this particular portion, which are repeated so many times, which is not coding for any protein, but it is forming a large portion, this sequence show a very high degree of polymorphism. Okay. Now, polymorphism is a variation at genetic level. Since DNA from every tissue, if we talk about the blood, hair follicles, skin, it can be bone, saliva, sperm, etc., from an individual, so a same degree of polymorphism means same number of copies will be observed, but become very useful identification tool in forensic application. Okay, so if we take any DNA sample from the blood, hair follicle, skin, bone, anything, it is going to be show the same degree of polymorphism, same variation, and that is a unique thing. But this variation will differ from the another individual. And this polymorphism or this variation which are present in a repetitive sequence in an individual, that is inheritable from the parent to the children. And because of this, DNA fingerprinting is also used in paternity testing in case of the parent disputes. So we can say the polymorphism or variation in DNA sequence is the basis of genetic mapping, is the basis of genetic mapping of human genome, as well as DNA fingerprinting. And this is essential that we understand that what DNA polymorphism means in a simple term. So polymorphism, which is a variation at a genetic level, which seen in case of which is seen or which variation is seen from individual to individual, that arises due to the mutation. And the allelic sequence variation has traditionally been described as a DNA polymorphism, or we can say allele sequence variation means variation in the nucleotide sequence of allele, that has been described as a DNA polymorphism. If more than one allele at a locus occur in human population with the frequency greater than 0.01, then it is known as polymorphism. Means the 0.01% more than 0.01% variation is seen, then it is considered as a new allele. So in simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred as a DNA polymorphism. So inheritable mutation, which can be inheritable from the one generation to the another generation, is observed in a population at high frequency. Means it is observed in so many individuals. Then it is referred as a DNA polymorphism. Polymorphism. Probability of such variation, which is very high, to be observed in non-coding DNA sequences. Be remember. First, you should know that what is DNA polymorphism. That if an inheritable inheritable mutation is observed a population in a population if an inheritable mutation is observed in a population at high frequency, it is referred as DNA polymorphism. Now this variation, it is known that the probability of presence of such variation to be observed in non-coding DNA sequences. DNA sequences. And it would be higher as mutation in these sequences may not have any immediate effect in an individual. So now it is also known that mutation in non-coding sequences may not have any may not have any immediate effect in an individual reproductive ability. So such mutation keep on accumulating generation after generation and form one of the bases of variability or polymorphism. So generation to generation, this variation or mutation accumulate, but it did not show a phenotypic change in the non-coding sequences. They get accumulated, and there is a variety of different types of polymorphism, ranging from a single nucleotide difference to a very large difference or very large scale change. And it is known that for evolution and speciation, this variation or accumulation of mutation or the polymorphism is very important. So we remember for evolution and speciation, that is formation of new species, such polymorphism play very important role. Okay, so this is the what a thing of DNA polymorphism. So now the differences or this variation is one of the important base of DNA fingerprinting. So now we can understand that the basis of DNA fingerprinting is the basis of DNA fingerprinting is VNTR. And VNTR is a variable number tentative repeat. And these are the obviously a repetitive DNA, so we can say it is a satellite DNA or repetitive DNA used as probe that show very high degree of polymorphism. Very high degree of polymorphism or variation from individual to individual. Now, and to identify this differences, this variation among individuals, a DNA fingerprinting technique was developed. So it is known that the technique of DNA fingerprinting was developed by Alex Jeffrey. So technique of DNA fingerprinting was developed by Alex Jeffrey. Okay. Okay. So now we are going to talk about the technique of this DNA fingerprinting, and we can say how by DNA fingerprinting we can compare the repetitive sequences or nucleotide sequence of two individual DNA, and we can make them and we can identify their unique sequences and make them particularly to different individual. So now it is known that this DNA fingerprinting is useful in some of the many things like paternity disputes, personal identification, and criminal cases. To solve some of the criminal cases, it can be used. So now here we can use this particular technique to solve the criminal case. Okay, and in criminal case we can solve this criminal case or paternity testing. So if we can say what are the practical application before understanding what its technique, how the fingerprinting is done, practical application, if we talk, the first one is paternity maternity to solve maternity paternity disputes. Means if one child is there and so many parents are claiming that this is our child, so at that time how we can solve, we can compare the DNA fingerprint of a particular or genetic sequence of the particular child with the different parents with whom it is exactly majorly it is the parent of this, he they are the parents of this particular individual. Second, to solve criminal cases and criminal and criminal identification and forensics. Third is personal identification. And fourth, close relation of immigrants. Okay, so for this all we can particularly use this practically, this DNA fingerprinting. So now we are going to take a one criminal case and we are going to solve this. So by and how this technique is going to be used. So steps of technique of DNA fingerprinting. Now see, if we are taking a one criminal case, and we are taking some one example. Suppose we have gone for some visit of any particular industrial area or any forest. Now at that forest, we have seen that the death of an individual will occur. And if suppose a death of an X individual is occurring and three individuals are running from that side, suppose A, B, and C, and we all have seen that particular crime in front of our eyes. Now police come and police verification comes and they will ask us question and we can say that the death of this X individual may be done by this A, B, and C individual. But might be this all are not are criminal. So if we want to solve the case and we have to identify that the criminal or murderer of that particular X individual of A, B, C is whom. So at that time, we will carry out the same technique of DNA fingerprinting with the DNA of X individual, the DNA of ABC, that is we can suspect. Okay. So first, what we'll do? The first is isolation of DNA. There is a technique of isolation of DNA. So in this, we will take one Appendorf proof or one Appendorf tube is there. This is like this. In this, we will take a sample of this particular X. In another tube, we will take a sample of, suppose in this we are taking a sample of X, then another tube we will take a sample of A, then we will take another in another tube sample of B, that is DNA, and another tube the sample of C. And here this DNA get This is a what a cell culture, and from this cell culture we are going to isolate a DNA. So DNA get extracted by the DNA isolation technique. Now when this DNA get extracted, then later on second step is done, that is a digestion of DNA by restriction endonucleases or enzymes. So here this DNA which is present in this get digested into small fragments like this by the restriction enzymes which we have added in this tube. After the restriction enzyme, the DNA segment we have taken from the X individual and the separate tubes of the A, B, and C individual, that all DNA in separate tubes of this all four individual has been digested. Now, after digestion, the third steps will be done with the all tubes. Okay, suppose this is having a culture. Now, suppose we have this is having a particularly restriction fragments of A individual. We are taking another tube. In this, we are taking a DNA fragment of the A individual. Another tube in which we are taking the isolated DNA, we are adding a restriction enzyme. So these are the fragments of B individual. And in another separate tube, there is a DNA fragments of C individual. Now all have been particularly digested, and now then we carry out a third step with all these tubes having a restriction enzyme digested fragments. And the third step we carry out, that is the separation of DNA fragments by electrophoresis. Now this electrophoresis is specifically known as agarose gel electrophoresis. Now what is done in this? In laboratory, there is a one instrument which carry out this particular agarose gel electrophoresis. And when we see the side view of this instrument, that look like this, and on this side you will see they have a particular negative electrode, and on this side they will have a positive electrode. And here there is a presence of casting tray. Now when we see, and in this casting tray up to this level we add agarose gel. Now agarose gel get particularly solidified, but here we put a comb which will create a wells in this agarose gel. So now when we see this agarose gel, its top view will look like this. This is the negative side, this is the positive side of electron. Here some wells are created, otherwise the overall this casting tray is with the agarose gel, but this gel is not present in these wells. Okay, and in these wells we keep a particularly fragmented DNA of X. In this we add a DNA of A, in this DNA of B, and in this DNA of C we add. Okay, and what we add? The extracted fragments. Now fragments which are added here, they can be of smaller size, they can be of larger size. And here if we see, this is the agarose gel which is somewhat thick. Okay, it is somewhat thick. So if this agarose gel is thick and it is made up of agarose gel has some porosity, has porosity. So whatever the fragment which are smaller than these pores of agarose in the gel, they can move further or they can move far away. So here you will see the presence of small fragments, and towards this side you will see the presence of what, a large size fragment. So small size fragment easily move through agarose gel towards positive electron, whereas the large sized DNA cannot move away from this. So towards the positive electrode, there are smaller sized DNA fragments are there, and towards the particularly we can say negative electrode, because why whales are created towards negative side, because DNA is negatively charged, and here it is a positive electrode, so DNA will move towards this positive electrode. Okay, so these fragments are moving, but these fragments which are in this particular agarose gel, they cannot be visualized, they are not visible, and these all particularly fragments of DNA which are present in the agarose gel, they are a single stranded. Now, what we carry out, the fourth step we carry out, and in fourth step, this particular, this DNA fragments, after separation in agarose gel, they are, the southern blotting is done. Now, what is southern blotting? So, in southern blotting, if we talk, in southern blotting, if we talk, the DNA fragments are transferred to synthetic membrane like nitrocellulose or nylon membrane. Okay, they may move towards that side, and this the DNA fragments are separated, which are transferred to the nylose membrane by a technique known as southern blotting. So what we do, we will take agarose gel, we will keep a nitrocellulose membrane, and over nitrocellulose membrane this agarose gel is kept, and then they are pressed and they are kept in one particular chamber where the agarose gel get liquefy, and that will go move out, and the DNA fragments will here remain attached like this. So like this is a X DNA fragments, A, this is B, this is C. Now here also they are not going to be visualized, they will be like this. Suppose we will observe like this segment in a later step, but they will not be visible on southern blotting or nitrocellulose membrane also. So now on this, the fifth step is carried out, that is hybridization using radioactive DNA probe. Now the question is, what is DNA probe? DNA probe is short sequence of DNA complementary to VNTR sequence of X individual. Now, if this DNA probe, which is a single stranded, okay, this is DNA probe is also single stranded, so it can bind with the single stranded sequence which are present in this on this nitrocellulose membrane, and that are complementary to the what VNTR sequence of X individual. So it can bind with the whatever the VNTR sequence is present in this DNA. Okay. Now, particularly if this is the X individual, see this is the sample actually. Here I am talking again X individual, but it is a sample. So if something we have found the hair follicle near about this particular what particular criminal case or a person X who died. Okay, this we can't call it as, we can call it as a sample. Okay, So if person X is died and near about one hair follicle we found or one blood sample we found which is not of that individual, then we can expect that might be this particular blood sample or hair follicle is of the person who have killed this X individual. So because of this X is this particular fragment, or with this X which I can say is sample, this is the DNA of that particular hair follicle blood sample which is found near about the criminal case or the particular person who died. And if this VNTR sequence are complementary to each other and they are binding to this one, then they will bind to the out of ABC to DNA sequence any one individual whose blood sample who died, who killed this particular individual, and whose blood sample was or hair follicle found near about the criminal case, at the location of criminal case. So this radioactive probe will bind to this X, this is sample DNA bands, and same binding will occur out of this ABC to one. And to whom the radioactive probe will bind, that will be the particular what individual or a criminal who killed this particular X individual. Okay, so when we carry out, we add this DNA probe, and here these are the wells. This is not wells because so here these are the its individual its bond now. So this will be radioactive and this will be visible when we add this particular probe, because probe are radioactive, so it is not visible here, these bands are not visible here on nitrocellulose membrane, but here bands, DNA bands are visible because of the radioactive probe. And it will be bind with the another sequence which are similar to this. Suppose this is a sequence similar to this in B, then here it can bind, here it can bind, there will not bind. But in B individual, might be the sample is of this B individual, then the same radioactivity will be observed in B individual. If same radioactivity is seen in B individual, then compared to the A and C, then this B is the obviously a criminal, and this is the sample. So means this sample for hair follicle or blood sample which is found near about the criminal case or location, it is of not a B individual that we can determine. But here these bands are radioactively, they can be visible. Bands are not actually visible. These bands will be visible in the next step. Will be visible in next step. So the next step is after binding of this probe to the specific DNA sequences of the sample and this person whose blood was this sample or hair are of this sample, then in a sixth step, excess of probe is washed off, okay, and the probe bind to the particularly DNA pattern of the membrane. And then in the seventh step, the detection of hybridized DNA fragments occur by placing X-ray film near nylon membrane to detect the radioactive pattern, and this is known as DNA fingerprints, because here the bands are visible. So as we have seen in the previous diagram, suppose this is the particular segment, like this, and DNA bands are like this, as we have seen in case of the previous one. This bands will be visible when X-ray film is placed nearby it. So this bands of sample are matching with the B individual, means B individual is the criminal. That can be sorted by this technique. So this is the whole technique of DNA fingerprinting, which tell us that how the particularly this DNA fingerprinting was developed, and such a way the criminal cases and maternity paternity testing can be maternity paternity disputes can be disputes can be solved. Okay, so this is about all technique. Now we have to note down some of the important points. Note point: we remember that the Alex Zafre developed the particular technique of this DNA fingerprinting, but it is known that Lalji Singh and VK Kashyap are Indian experts, are Indian expert in the field of DNA fingerprinting. Okay. Now, one note point that you should also remember, that a definition sometimes they ask, that is cDNA. cDNA is complementary DNA. This is for just for information. And this complementary DNA, it is a synthetic type of DNA generated from mRNA okay, by reverse transcription. So here the scientists use this enzymatic reaction and convert this information which is present in mRNA back to the DNA, and that DNA will be known as cDNA. And this cDNA is then cloned, and the cDNA library is formed, which has a cDNA complementary DNA of many mRNAs. Okay, so this is all about the DNA fingerprinting and this note point. I hope you understood well, and this is the end of this molecular basis of inheritance chapter. I hope you understood well. Thank you.